So here is the introduction. We did too much. Now let's talk about something about health and lose your weight. Here's our star bitter melon, a tropical plant, has been shown to have many functions, something like anti-diabetes, antiviral, and anti-neuroplastic effects on the human body. It has been thought to improve the activity of human gut microbiome community, which is composed mostly anaerobic bacteria that regulate energy, metabolism, and immunity. The human gut microbiota can also act as a trigger for low-grade inflammation, a characteristic of both obesity and other diseases like diabetes. So the next one. I thought we eat too much high-fat diets in this lunch. We need more fibers. So the high-fat diets can change the composition of gut microbiota. Something decreases like bifidobacterium. It can protect our gut barrier. Some compounds like LPS, also called endotoxin, endotoxin increase. So endotoxin enters the bloodstream. It can touch and bind the macrophage surface. So the macrophage surface can induce the inflammation, and which is a major factor in obesity and other metabolic disease. So there is two persons. It's a happy but it's fat man and a very pretty and slim girl. So the bad thing is endotoxin. Endotoxin can contribute to the metabolic abnormality. So particularly something about obesity and diabetes. While short chain fatty acids in Chinese is 短链脂肪酸, can protect our body from inflammation, so block the inflammatory pathway. You can come to a little slim girl like she.
Sorry, I didn't want to put a real person up there because I thought it'd be too mean.
It's like me. It's better. In our experience experiment by our lab and other students, so he found that a bacterium, Faecalibacterium, one of the bacterium can produce butyrate. Is a type of the short chain fatty acid that's essential for the intestinal cell. So is our PI, Professor Zhao Liping. He used himself as the subject of one of his tests. You can see two pictures. The picture before he do the test is a little more bad, I think. But after 2 years, he lose 20 kilograms. 20 kilograms is very much. So you can hear it's very, I'm not sure it's slim, but it's healthy. Yeah, healthy. This is bitter melon, and this is Chinese jam. It's major. I think the major composition of this medicine or something like you must be sure that the regimen. Here is the report published in Science just 2 months ago. It shows that a lot of successful experiments in our lab and in our Professor Zhao. So let's talk about something before our experiments, previous experiments. Our experiment is a really continuation of Stone's work. He worked our lab here as an undergraduate. He concludes that the bitter melon indeed, in fact, influences the composition and activity of the gut microbiota. So he identified 8 PCR DDG bands, and 7 of them, the enriched band, were short-chain fatty acids producing bacteria, and the last one, the love, inhibited the band belong to endotoxin. These are harmful compounds. Endotoxin produce bacteria. So, our experiment objective were split 2 parts. One is the real-time PCR. Another is gas chromatography. We use RT-PCR quantitatively verifying that the bitter melon increases the short-chain fatty acids producing bacteria and inhibits the endotoxin producing bacteria. And about 1 month later, they use gas chromatography to separate our compounds of the fecal sample supernatant. So we can conclude that whether or not bitter melon influences the intestinal fermentation. So let's welcome to Chris.
I think we're running short on time, so I'm going to go through these really fast. If you want to see them in detail, find me later. So Sloan made the different conditions by mixing 5 different concentrations and 5 different incubation times. We had 0, 2.75, 7.5, 15, and 30 milligrams per milliliter of bitter melon, and 0, 6, 12, 24, and 48-hour incubation time. He also made the 3 biological replicates, A001, 2, and 3. And when we made the plates for RT-PCR, we made the 3 technical replicates. So RT-PCR quantified 5 specific bacteria and the general all bacteria. We used the 16S rRNA gene for most, except butyrate-producing bacteria, which was the butyrate coenzyme A transferase gene, and sulfate-producing bacteria, which is the adenylene 5-phosphorsulfate reductase gene. And so we began with total bacteria. When we measured it, we did absolute quantification and relative quantification for the specific one. In absolute quantification, we compare the copy numbers, which is just the measure of the bacteria, and we divide it by the total amount of DNA, which is measured in nanograms. So one of our main focuses is particularly bacterial pregnancy. This is the SCFA producing bacteria. As you can see, it increased on the 30 milligrams per milliliter, shows a significant difference from the 0 milligrams per milliliter of bitter melon. Our next one is butyrate-producing bacteria, which is also another short-chain fatty acid producing bacteria. And prior to the 12-hour mark, it increased drastically, but afterwards it declined. And our final SCFA-producing bacteria is bifidobacterium. The results weren't what we expected. We had hoped that bitter melon would increase this, since this is an SCFA-producing bacteria. This could be because it might not have an important role in producing SCFAs. And as of right now, we aren't exactly sure what its role is in the gut. Our first endotoxin-producing bacteria is sulfate-reducing bacteria. It showed that after the addition of bitter melon, the count reduced significantly. And our final bacteria is Enterobacteriaceae, which has a much clearer and prettier graph. As you can see, the 30 milligram and the 15 has significantly less bacteria than the 0. So to summarize, Fecalibacterium pregnancy and butyrate-producing bacteria both increase the concentration of bitter melon. Bifidobacterium showed slight inhibition, and SRB and Enterobacteriaceae both decreased with the addition of bitter melon.
My turn. So the SOM's EGG PCR and it is 454 viral sequencing, and the before RT-PCR is structure and analyze. So we use gas chromatography to do something about the function. So here is the total ionic chromatography. We can see this picture. This is astatic acid. This is pyro. I don't know. I'm not sure. I check it. It's propionic acid. This is isobutyrate. This is butyrate. This is isovaleric acid. This is valeric acid. In Chinese, you can say that 乙酸、丙酸、异丁酸、丁酸、异戊酸、戊酸. You can also call it C2, C3, iso C4, C4, iso C5, C5. So there is two different equivalent concentration, 18, 20 by 1.25 milligram per milliliter. So we use this to create a standard curve to identify the time of a period for the different components in the samples. So this is our standard curve. Our results were adjusted by the internal standard and the distributed rate of the target structuring fatty acids in the ether and water solution. So the standard curve shows that each element has R squares greater than the 0.980. So we'll use this curve to convert the peak height into the concentration in the sample. The first acid, astatic acid, we can see here the significant increase in astatic acid level from the 0 to the 18 milligram per milliliter betamalin. So we can see the high concentration of the betamalin, the high level of the astatic acid. Oh, sorry. The right picture is 24 hours marks box graph. We can repeat the 24 hours to show how difference between the two different concentration betamalin. The next acid, C3. This picture shows there is no significant difference between the 0 and 18 on other concentrations. So we don't think it's a difference by influence by the betamalin. The isobutyrate acid, there is too much 0 because I think the concentration of this acid is too low for the machine to read. So we label them to no data. We label them to the 0. The butyrate acid is an important acid we research. We can see from the picture there is a very significant difference between the 0 and the 18. We use the ANOVA test for the significant difference. So we can see from this picture. They are also dependent on the betamalin concentration with the high level of the acid. The isovaleric acid and the valeric acid is C5. With the higher concentration of the betamalin, two acids seem the same trend. To show the same trend, the more concentration of the betamalin, the low level of these two acids. So we sum six acids to the total short chain fatty acid. From this graph, we can see the more with the high concentration of the bitter melon, the high level of the total short chain fatty acids. So their summary from the six graph, we can see that the acidic and uric acid, two major short chain fatty acids, show the greatest concentration dependent increases. Valeric and isovaleric acid decreases after adding the bitter melon. The total short chain fatty acids increase with the concentration of the bitter melon, supporting the sports we get from the RT-PCR.
So RT-PCR and mass chromatography both analyze the gut, one through structure and the other through function. When the RT-PCR data showed the increase in uric-producing bacteria, then the gas chromatography data also showed an increase in uric, as you can see here. So they coincide with each other. Because the data are very similar, we can conclude more simply that the addition of bitter melon does in fact enhance SCFA-producing bacteria as well as SCFA products and inhibits endotoxin-producing bacteria. Our results have show that bitter melon can control the gut microbiota and alter it to benefit health, as you can see because of these three. And hopefully in the future we can use bitter melon to suppress inflammation and help diabetes and obesity. But as of right now, we've only tested the effects of bitter melon on samples taken from a healthy young individual. So in the future we would hope to do similar tests on samples from diabetic or obese patients and hopefully come to the same conclusion. And thank you everyone for making this summer a success, and yeah, we're out of time.
Thanks for your excellent work. Professor Zhao is 55 years to lose 50 pounds. I think you girls like a street life lose 20 pounds in the summer. So can you, what's the secret in behind for amazing performance?
Well, it's actually there's no secret. It's a diet now, and it's a combination of whole grains, Chinese yam, and peach melon. We select peach melon for peach food work is because the inspiration from traditional medicine. Because peach melon is bitter, obviously, and there are many Chinese medicine which are bitter. And then Chinese doctors believe that bitter medicine or bitter food can quench the fire within your body. That's their theory. And so now we figure that the fire within our body probably is somehow related with inflammation. So all those medicine or medicinal foods which are bitter probably can somehow help us reduce our inflammation. In our theory, by changing gut microbiota. And so that's why we started the preliminary test along this line. But we still have a long way to go to figure out the exact mechanism. But we just published a paper on a compound called berberine. So if you are interested, you can Google and then get the paper because it's published in PLOS ONE. So it's free. Okay? And we did a very systematic analysis of how this compound change gut microbiota and prevent obesity and diabetes from animals that have that diet. So it's good for now. Yeah.
How does inflammation okay, so okay, so Joseph he'll talk about that's part of his experiment this summer. Oh no, okay, so what happens happened was they took the sample, fecal samples from him, and then added, yeah,
It's an in vitro.
yeah, in vitro cultivation.
So would it be clearly different if the viron had already been processed by the stomach and
Yeah. Like that would be, I think, the ideal way of doing it, but it's hard to control diet, like making someone just eat bitter melon, you know, like no pizza, no soda, no nothing else. Yeah. Okay, I think we're out of time.
yeah. One or two questions on the
If you have questions, write them down, and then you can come find our young scientists after and ask them.